Hi, everybody. This is Mrs. Stern again, and we're going to take a minute to put together some of the things that you've learned about different organelles, particularly the endoplasmic reticulum, the Golgi apparatus, and the vesicles, to help us better understand how cells transport and export proteins. So remember that proteins do all the jobs in our cells, and we've talked about enzymes, and we've talked about protein channels and protein pumps with facilitated diffusion and active transport. So cells are constantly making proteins in order to do all these jobs. Proteins are basically the machinery of the cell. And so a lot of the organelles that you've learned about work together to play this important role of manufacturing, moving, and distributing all of this machinery to all the places that it needs to go. So if you look in this picture, these arrows are showing like different places that proteins can move, and we'll talk about all of this, but you can see some of them stay within the cell. Some cells also have to send proteins out, and an example of that would be insulin. So in this picture, these purple dots could represent insulin. Insulin is a hormone that regulates your blood sugar. Your pancreas produces it. And so the cells of your pancreas have to have a way to get those insulin proteins out of the cell. So we're going to use that as an example just to help us understand what's going on here. So the journey of a protein begins from the ribosomes. In this picture, the ribosomes are the blue dots. Ribosomes are the ones who actually make the proteins. Ribosomes basically provide a place where amino acids can come and get linked up to make a chain. We will go into much more detail about that in a future unit. Ribosomes, in order to make proteins, need to get instructions, and those instructions come from the nucleus. So there's this kind of relationship between the nucleus holding the instructions, which is the DNA. DNA is basically just a protein recipe book. So there's instructions in the nucleus. The instructions get sent to the ribosomes, and the ribosomes make the protein. We'll cover that at a different time, but now the proteins have been made, and the question is, where do they need to go, and why, and how do they get there? Okay. So you can take these notes on the back of your packet or in that blank page, but we're going to talk through the various steps of where the protein would go. Okay, I'll use a little purple dot since we have purple insulin coming out there at the end. So here's our insulin protein, and it's in the ER right now. First, that insulin protein is going to need to travel kind of through and between these various layers of the endoplasmic reticulum. And there may be places where it's kind of moving around within these sacs, because really the endoplasmic reticulum is just these like folded sacs. But that's the first piece. So the proteins will move through the ER. Okay, so once the proteins have moved through the ER, they'll eventually kind of get to this outer part, and you can see here is a vesicle coming off of the ER, because all of these organelles have their own phospholipid bilayer membrane around them. That's all of this. That's what this is. It's just phospholipid bilayer. So a little piece of that membrane can sort of bleb off and carry those proteins with it. And that's what's happening next. So when the protein is kind of done moving through the ER, it's going to go into a vesicle, and a vesicle will form. A vesicle forms off of the ER. And carries proteins to the Golgi apparatus. The Golgi apparatus is named for the Italian scientist who discovered it. Okay, and so then the Golgi apparatus, or here it's labeled the Golgi complex, different scientists call it different things, is the orange here in the picture. And again, it's like kind of flattened sacs of membrane, sort of like discs almost, with open space within them. So when the vesicle gets to the Golgi, the vesicle will merge with the Golgi apparatus and dump out its contents into the Golgi. As the proteins move through the Golgi apparatus, they can end up getting modified. What that means, before we write that down, is like some proteins in the cell membrane, for example, have carbohydrates attached to them. So in this picture, that's the protein part. That's what would be coming from the endoplasmic reticulum. But in order for the protein to perform its job in the cell membrane, it needs a little piece of carbohydrate attached to it. So the Golgi apparatus would be the place where that gets attached to the protein. Okay. So proteins are modified, and sometimes proteins need to, like two proteins might need to join together to make a bigger protein complex. That could happen in the Golgi apparatus as well. So they kind of get like modified and finished if there's anything about their shape that needs to get finished. That would happen in the Golgi. So proteins get modified and finished in the Golgi. So by the time a protein moves through the Golgi apparatus, it is now fully ready to do its job if it has any things it needs to have attached or changed about its shape. So you can see coming out of the Golgi apparatus in this picture, there's a few different arrows. We're going to follow this arrow because we were talking about the insulin proteins and how they would get sent out of the cell. So notice there's another vesicle here. That's what that is. And you can see the Golgi apparatus almost looks like it has little buds coming off of it. The artist is trying to show vesicles coming off of the Golgi and going different places. So this vesicle is going to carry those proteins to the surface of the cell. So a vesicle will come off of the Golgi I'm just going to abbreviate Golgi apparatus, and goes to the cell surface. And then once that vesicle gets there, then basically exocytosis happens. You remember exocytosis from when we talked about cell transport? Okay. And so then that vesicle will merge with the cell membrane and just dump its contents out. Another thing that could happen here is, like, if you think about the protein channels trying to get put into the cell membrane, this is also how protein channels end up in the cell membrane. If there was we'll zoom in on this vesicle. So if the cell made, if the ribosomes had made protein channels, those protein channels would be like embedded in the vesicle here. And then when the vesicle gets to the surface, the protein channels would just like end up in the cell membrane, and that's how they get there. So different proteins will travel around in these vesicles or on the vesicles kind of in different ways. All right, if you need to rewind to see any of my notes again, you can. Make sure you have those steps written down. And then I just want to briefly talk about other things that are happening here. So you can see with this arrow that really the cell is forming lysosomes. Remember that lysosomes are responsible for digesting stuff. Like they could be digesting wastes or things that the cell wants to recycle, like used up cell parts and stuff like that. Lysosomes have enzymes inside them. And so it might be that, the proteins coming here from the ER could be enzymes to make a lysosome. And essentially lysosomes are formed by this protein transport process. And then the lysosomes are like a special kind of vesicle that comes off of the Golgi apparatus. And then in this picture, it's showing a lysosome being formed here. And this vesicle could be coming from endocytosis. And so if the cell brings something in, it could take the stuff that it brought in and fuse it with a lysosome, and then the lysosome could help break it down. So hopefully what you can see is that there's a lot of different places a protein can go from after it gets made by a ribosome, depending on what that protein is for. But you can see how these different organelles are working together to help move those proteins around or send them out of the cell if that's something that if the protein is like insulin, it needs to get sent out of the cell. So go ahead and rewind if you need to watch any part again. Otherwise, you are ready to move on to the next step.